Today we're going to be discussing plasma membranes and solutions and the role of different types of atoms and molecules in those solutions. So let's go ahead and begin with plasma membranes. Now, plasma membranes are found throughout cells. For example, the cell membrane is a plasma membrane, nuclear envelope, endoplasmic reticulum, Golgi apparatus, the membranes around and within mitochondria and so on are all examples of membranes. So membranes essentially are packaging materials for cells. You can kind of think of it as the cellophane or transparent wrap or the baggies of cells. So let's go ahead and look then at the actual structure of plasma membranes. So the basic structure of a plasma membrane, and this goes for any plasma membrane, whether we're talking about advanced organisms like ourselves or primitive organisms like bacteria, is that it is a double phospholipid layer, so-called biphospholipid layer. And we talked a little bit about those before, with different kinds of proteins embedded in that phospholipid layer. There'll be some other molecules as well, like cholesterol is also embedded in here, but this primary structure, the basic idea I want you to get about a plasma membrane is that a plasma membrane is composed of a biphospholipid layer with proteins embedded. Now, again, there are some other things like other proteins in the extracellular matrix or maybe carbohydrates out here, also some additional proteins in the intracellular material, but the membrane itself, biphospholipid layer with proteins embedded. Now, these proteins will have a number of functions. Some of them are transport molecules, pore proteins, some are enzymes, receptors, and so on. But we're going to be most interested in these kind of molecules here that allow for passage of materials across membranes. Now, plasma membranes are described as being semi-permeable or selectively permeable, and I'll be abbreviating that as SPM. And so what that means is that some things can pass through, but some things cannot pass through. So, again, we have our double layer of phospholipids, a biphospholipid layer, hydrophilic heads, hydrophobic tails. And again, between the two, it's a fairly effective barrier, particularly these hydrophobic tails. Now, particles that are lipid soluble can generally pass through the membranes, at least if they're not too big. Smaller particles, it's like gases here, oxygen can pass through the membrane fairly easily. Polar molecules, though, have a much more difficult time. Things that are water soluble have trouble getting through the lipid barrier, and so they cannot pass through the membrane. So they require either transport molecules or pore proteins that have openings in the membrane to allow them to pass through. So again, some things can pass through, other things cannot pass through. That makes it a semi-permeable barrier. And so chemical properties of these particles as well as size has a lot to do with whether they can pass through the membrane or not. Okay, now when we talk about cells, and even sometimes extracellular material like blood plasma, things like that, we're really talking about solutions. And so a solution is a homogeneous mixture of ions or molecules of two or more substances. And really, when we talk about solutions, we're talking about gases or liquids. And we're really going to be most interested in liquid solutions, solutions that have some kind of a liquid component to them. And so when we look at a solution then, we describe the substances as being solutes, which will be the relatively less abundant material or materials, and the solvent will be the most abundant or relatively more abundant substance. So again, we're normally going to be considering the solvent as some sort of liquid and the solute as some sort of solid or perhaps gases that are dissolved in that liquid. In particular, when we're talking about solutions of life, we're talking about aqueous solutions. And so the prefix aqueous is telling us water, so the solutes are typically dissolved solids or gases. And again, in living solutions, when we're talking life, we're talking about the solvent being water. So aqueous solutions, when we talk about aqueous solutions, and we normally just define them by the solute concentration. And so what I mean by that is that I might tell you that we're working with a 5% starch solution. Well, 5% starch solution, I'm telling you that it's 5% starch, but what I'm not telling you is that it's 95% water. So again, the assumption is any solution we talk about is an aqueous solution. I might tell you that a solution is 0.7% saline. Well, what I'm saying there is that it's 0.7% salt. And again, what's unstated in that definition is that since it's an aqueous solution, it would be 99.3% water. So always remember that whenever I talk about a solution, what's unstated is the concentration of the water. Now, diffusion is a property of particles in solution. So it is the movement of particles in solution from areas of higher concentration to lower concentration. And that's a concept that you have to continue to remember as we talk about solutions. Particles will always diffuse or move from where they're more concentrated to where they're less concentrated. And it doesn't matter whether we're talking about a solute particle or whether we're talking about water molecules. They all diffuse from where they are to where they are not. So let's get a couple of examples. Now, what I want to show you this just shows us kind of the frenetic quality of solutions. So this is representing actually a plasma membrane, a very realistic example. Here's our biphospholipid layer, the water-soluble heads, the fatty acid tails, and so on here. And these are showing us some proteins, pore proteins that allow water to pass through the membrane. And again, what's important about this is I just want you to get the idea of movement. So these water molecules are colliding with one another, and that's what diffusion is all about, just movement of particles. So particles have an inherent kinetic energy of movement. And so again, I'm going to kind of keep this going for a second so you get a sense of all the collisions that are occurring. So any particle in an aqueous solution then is constantly being bombarded and bounced around by the surrounding water molecules. So when we look at diffusion then, diffusion is described as being passive. It doesn't mean there's not a lot of energy there, but it's kinetic energy, the energy of movement. So when we say it's passive, it means that the cell does not have to necessarily add energy. No ATP is consumed in this. Now, particles will always follow their concentration gradient in diffusion. So they go from where they're more concentrated to where they're less concentrated. And particles eventually disperse themselves equally in a solution, reaching a state of equilibrium. All right, so let's look at a couple of examples of this. One, we're actually showing a semi-permeable membrane here, but we have particles that are in a solution, more concentrated on this side than this side. And so the particles then follow their concentration gradient and move from where they are more concentrated to where they're less concentrated, and eventually will reach an equilibrium of sorts. I also want to show you an example of this using some crystals of dye known as crystal violet. And so what I'm going to do is put some of these violet crystals in some water, and I want you to see what happens when we do that. So initially you can see these particles will just start out on the top of the surface, a few are dropping down. But the crystals, of course, are where the dye is most concentrated. They are slowly dissolving into that solution. And water molecules will eventually bombard them. They will collide with one another and eventually be dispersed throughout the entire solution. And if you look at this beaker right next to it, just a short time ago, I did the same thing. And you can see now that those dye particles have completely dispersed throughout the solution. And I did not stir that solution or in any way agitate that solution. That was purely through diffusion, dispersing those particles. So that is another aspect of solutions that I think is important, is that the particles then will disperse themselves and are not pulled out of the solution by gravity. So they basically can stay in suspension in spite of the gravity trying to pull them down. Okay, now I want to next consider some special cases of diffusion. One of these special cases of diffusion is osmosis. So osmosis is the diffusion of water through a semi-permeable membrane. And water, as will any particle, always follows its concentration gradient. Now, when we're talking about solutions separated by membranes, there's always a comparison that we are going to be involved with. We're going to describe one solution relative to the other. And I'll describe this as tonicity. So I may be asking you, or you may hear people talk about the tonicity of a solution. And when we say that, we're actually comparing two solutions separated by a plasma membrane. So one term that we use is a hypertonic solution. A hypertonic solution would be on one side of a plasma membrane or a semi-permeable membrane. And a hypertonic solution has a greater osmotic pressure. What that means is that it has a greater capacity to pull water towards it. Hypertonic solutions gain water. So this is an important concept. When we talk hypertonic solutions, I need you to think of it as a chemical sponge. So just as you can take a dry sponge, you put it in water, it will suck up that water. The same is true in solutions. Some solutions have a greater capacity to draw water than others. So the greater the osmotic pressure, the stronger it is as a sponge, the more it will suck up water and the more water it will gain. So again, a lot of this is hard to remember, but I don't care how you remember it, but I do need you to know the hypertonic solutions gain water. They are chemical sponges that are going to suck up water. Now additionally, hypertonic solutions generally have a greater solute concentration. So if we were comparing a 2% sucrose to a 5% sucrose to 5% sucrose, we would describe as being hypertonic. And again, we're going to look at some examples in a second here. So now, if one solution is hypertonic, the solution on the other side of the membrane then we would describe as hypotonic. And again, this suffix tonicity you can think of as thirst. So a hypertonic solution is hyperthirsty or very thirsty. A hypotonic solution is less thirsty. So a hypotonic solution would have a lesser osmotic pressure compared to a hypertonic solution. It would lose water to a hypertonic solution. And generally they have lower solute concentrations than a hypertonic solution, at least if we're comparing the same type of molecule. Now, also as we mentioned earlier, as I discussed earlier, particles eventually reach an equilibrium. They disperse themselves equally. And if we have two solutions separated by a semipermeable membrane that have the same solute concentrations, we call those isotonic solutions. So isotonic solutions have an equal osmotic pressure, so they have an equal capacity to draw water. So they exhibit no net gain or loss of water. And they generally have, as I mentioned, the same solute concentration. And because they have the same solute concentration, they would also have the same water concentrations. All right, so let's go ahead and look at an example of osmosis, or potentially how osmosis might work. We have two solutions in a U-shaped tube separated by a semipermeable membrane. So one of these is a 2% sucrose solution, and the other side of the membrane we have a 6% sucrose solution. So the 6% sucrose solution has a greater solute concentration, and this is the hypertonic solution. Okay? The 2% sucrose solution has a lesser solute concentration, and this is the hypotonic solution. Now, the other side of the coin then, if this is 2% sucrose, it's 98% water. If this is 6% sucrose, it's 94% water. So we're assuming here that the sucrose cannot get across the membrane, but that water can pass through this membrane. So water, as any other substance will do, will follow its concentration gradient. So because there is less water on this side than this side, water will diffuse from the hypotonic solution into the hypertonic solution because it's hyperthirsty, it's acting as a sponge. And then as that happens then, as water is removed from this solution, these particles become concentrated. And as water is added to this side, these particles become more diluted. So initially this was the hypertonic solution, less water. Hypotonic solution, more water. So water diffused from where it was more concentrated to where it was less concentrated. But eventually, because of the change in water volume, the concentrations will be equal, osmotic pressures are equal, and we would now describe these two solutions as isotonic. Let's look at another example. Let's say we have a red blood cell, 0% 7 NaCl. And in the blood plasma, let's say the blood plasma is 1.7 saline NaCl. Well, if that's the case, this would be the hypotonic solution. The cytoplasm of the red blood cell would be hypotonic. The blood plasma would be hypertonic. So again, keeping in mind that if this is 0.7% saline, it's 99.3% water. If this is 1.7% saline, it's 98.3% water. So water is less concentrated in the plasma than within the cytoplasm. So where is water going to go? Hopefully you pointed in that direction. So water is going to diffuse towards the hypertonic solution, greater osmotic pressure. It's acting as the chemical sponge. So what will be the consequence of that? Well, the red blood cell will shrivel, and that will decrease surface area. It won't function as well, potentially. Also, the same kind of thing could happen in a plant cell, and all that would happen there is that the cell would lose some of the fluid inside, and I'll show you in a couple of minutes how that would result in a plant. But basically the cell wall, plants have a cell wall. The cell wall will not completely collapse, but the cell membrane will peel back from the cell wall, and it will lose some of the fluid pressure inside of the cell. Okay, let's look at another example. What if the cytoplasm of the red blood cell is 0.7% NaCl and the plasma was 0.2% NaCl? So now, which solution is hypotonic? Which solution is hypertonic? Hopefully you answered. The cytoplasm of the red blood cell is hypertonic because of greater solute concentration, less water. Hypotonic, lower solute concentration, more water. And in which direction will water diffuse? Or osmose, I should say. It will follow its concentration gradient into the red blood cell. And the consequence of that would be that the red blood cell would expand, but eventually rupture. And that, of course, would not be a good thing because you need red blood cells to carry your oxygen. Now, again, in a plant cell, if the cytoplasm was hypertonic, water would move in, but it really won't rupture the cell wall. So what I would actually like to do is show you the difference in how plants respond to a hypertonic and a hypotonic environment. So here I have some lettuce. I've got one bowl here with lettuce in salt water. And so when I pick this lettuce up, you can notice how flaccid it is, how limp it is. So the water has been sucked out of the lettuce. So the cytoplasm of the lettuce then is hypotonic, and the salt water around it is hypertonic. You compare that to lettuce that I have in distilled water, 100% water. Here the cytoplasm of the lettuce was hypertonic, so water has moved into the plant cells, and so it's very crisp and it's very rigid. So in herbaceous plants, non-woody plants, water pressure within the plant, helps, hold the plant up. So again, if we compare the two, you've got a very flaccid plant where the water has been removed, and a very rigid leaf where water has moved in. So if you're trying to, crisp up your lettuce for guests or something like that, you definitely don't want to put it in salt water because that'll suck all the water out. Okay, now, the ideal situation, of course, if we look at the red blood cell example again, would be that both the cytoplasm and the plasma have the same solute concentration, they would have the same tonicity, they would be isotonic, there would be no net movement of water, water would be moving in equal quantities in both directions. And of course, that would maintain the shape of a red blood cell. Okay. Now, another special case of diffusion is dialysis, also called chemiosmosis. And this is diffusion of solute particles through a semipermeable membrane. And so solute particles, just as any other particle, will follow their concentration gradient when they diffuse. So again, looking at an example here, we have solute particles concentrated on one side of a semipermeable membrane. They will diffuse from where they're more concentrated to where they're less concentrated, as we're seeing here, and eventually will reach an equilibrium state, at least will attempt to reach an equilibrium state. Now, you may notice that this solution then would be hypertonic to this solution, and so water would be moving in the opposite direction. And so you might be wondering, can solute particles diffuse in one direction while water is diffusing in the other? In other words, can dialysis go in one direction, osmosis in the other direction? And the answer is yes, that can happen. It just means that they equalize more rapidly. You could also have different solute particles dialysizing in opposite directions. So here our orangish particles will follow their concentration moving from left to right. The purple particles will diffuse from right to left. And so again, is it possible to have particles moving in opposite directions? Yes, and they will eventually reach an equilibrium state. Let's look at still another example where we're now looking at a membrane that is permeable to both water and the solute particles. So we have a bag surrounded by dialysis tubing that is 3% sucrose inside the bag and 2% glucose, and that would also mean 95% water. And then in the solution outside the bag, we have 1% sucrose, 1% glucose, 1% fructose, leaving 97% water. So if we've got 1% sucrose outside the bag, 3% sucrose inside the bag, what would sucrose do? In which way would it dialysize? Hopefully you said from the bag to the surrounding solution. If we have 1% glucose outside the bag, 2% glucose inside the bag, in which direction would glucose dialysize? Hopefully you said from inside to out. If we're looking at 0% fructose in the bag, 1% fructose outside the bag, again, fructose will follow its concentration gradient, in this case into the bag. And now if we look at osmosis, 95% water inside the bag, 97% outside the bag, water will osmose into the bag. So again, we're following the same basic concepts of diffusion, follow concentration gradients. Now, there are other ways that particles get through membranes, and one of these is facilitated or passive transport. And so this may involve a channel protein or some type of a transport protein to get materials across a membrane. But again, because it says facilitated or passive, that means no ATP, no cellular energy is consumed in this. But if it is a passive process, particles have to follow their concentration gradient. So again, particles would diffuse from areas of higher concentration through a pore to where they're less concentrated. This would just be a pore protein. Now there may be specific types of transport molecules that bind only particles of a specific shape and then change their shape in response to that to deliver to the other side. But since no cellular energy is being consumed, again, they can only diffuse from where they're more concentrated to where they're less concentrated. Now, active processes consume cellular energy. They consume a molecule called ATP. And because energy is being added to the system, it is possible to oppose a concentration gradient in active processes. So one of these we've talked about before, active transport. And so let's look at an example of active transport. So we have a transport molecule. In this case, it's going to bind sodiums and take them across the membrane. And the change in shape of the protein will require energy. It will require ATP to be added to that. Now, because energy is being added to the system again, they can oppose a concentration gradient. And so now it will bind potassium and move it back across. And this is what's known as a sodium-potassium pump. It's crucial for our nerve impulses. So active processes then can oppose concentration gradients and take particles from where they're less concentrated to where they're more concentrated. And that's a very important characteristic. But again, takes energy to do that. Now, there are other ways to get materials in and out of cells. Not necessarily across membranes, but at least in and out of cells. And this usually involves larger particles. So one of these is known as endocytosis. So we're going to be taking particles into a cell. And one example of this is phagocytosis, described as cell eating, and the other is pinocytosis, which is described as cell drinking. So let's go ahead and look at an example of this. Here's our plasma membrane. Here's a large particle which cannot fit through some kind of a pore. And you'll see that the membrane actually will surround it and bring it into the cell within a membrane. Pinocytosis generally does the same kind of thing, but involves smaller particles. Okay, now, exocytosis is really just the opposite process. Particles will be taken from the inside of the cell and spit out of the cell, in effect. So let's go ahead and look at this example. So we have particles within a vesicle. The vesicle is going to bind the plasma membrane and spew those particles outside the cell. So again, this generally involves particles that are being packaged for secretion outside the cell, or they may be particles that are just too big to fit through a pore protein. Now, osmotic pressure, remember, is the capacity to draw water. And so some of the things that can affect osmotic pressure include solute concentration. So generally speaking, whichever solution has a greater solute concentration draws more water towards it. However, the number of electrolytes formed in a solution can affect that. So for example, if we look at sodium chloride, when it dissociates, you get a single sodium and a single chlorine, only two particles when that goes into a solution. If you take calcium chloride, calcium separates into one calcium and two chlorines, 3 particles. So if you had a 1% solution of calcium chloride and a 1% solution of sodium chloride separated by a semipermeable membrane, the calcium chloride solution would be hypertonic because it would actually have more particles in the solution than would the sodium chloride solution. So that's one of the factors that can affect osmotic pressure. Also particle mass. So if we were to compare sucrose to glucose, sucrose has a greater mass than glucose. As a result, the greater the mass, the greater the osmotic pressure. So a 1% sucrose solution then would have a greater osmotic pressure than a 1% glucose solution. And then particle charge can also affect movement of particles. That's a little bit harder to predict, but sometimes positive or negative ions have a better chance of getting across a particular type of membrane. So that's, again, a little harder to predict. And then other factors that can affect the speed of diffusion. One certainly is temperature. Generally speaking, as temperature increases, so does the rate of diffusion. Again, particle mass. The heavier a particle, the more slowly it diffuses. Or conversely, the lighter a particle, the faster it diffuses. And then third, the concentration gradient. So again, if you have two particles separated, especially across a semi-permeable membrane, the greater the difference in the concentrations, the faster the rate of diffusion. So a 1% and 2% solution, you'll have slower diffusion as opposed to a 1% and a 10% solution. Diffusion will be much, much greater. Now, I've been throwing around some terms quite a bit, and I may have used these more than once, but I want to quickly go through the term polar and nonpolar and synonyms for those. So we understand, hopefully, the idea of a polar molecule, but polar molecules are water soluble. That's another way of saying they are fat insoluble, meaning they will not dissolve in fats. They are hydrophilic, meaning that they like water. So hydro means water, philic means to like. So if something is polar, it's water soluble, means dissolves in water, it will not dissolve in fats. It loves water. And lipophobic is also saying that it's afraid of lipids, so it will not dissolve in fats. And then conversely, synonyms for nonpolar would be, if something is nonpolar, it does not dissolve in water very well. It's water insoluble. That means that it is fat soluble, will dissolve in fats. Means that it is afraid of water, hydrophobic, does not dissolve in water very well. But lipophilic, loves fats, meaning it does dissolve in fats readily. So again, different authors will use different terms. I wish I could tell you one is used more than another, but they're all used quite a bit. So hopefully you have a better sense then of what a solution is and how and why particles move within solutions. So until the next time.